friends, Happy New Year. Today I want to ask for your help in financing my anti-aging research project. The project aims at preventing age-associated cell dysfunction and death in the brain. Nothing impossibly bold, but we have a decent chance to delay the onset of dementias and allow people to remain themselves to the very end of their natural lifespans. The purpose is to move dementias to the back of the line of diseases waiting to kill us by confronting an independent mechanism of brain aging that can be relatively easily neutralized. The target is mutations or accumulation of mutations in mitochondrial DNA, which lead to inability of mitochondria to utilize oxygen and produce enough biofuel called ATP. As a result, such cells go dark because they enter a state of irreversible energy crisis and therefore cannot function properly. For lay audience, here's some background information. Mitochondria are our power plants, supplying energy to most biochemical reactions by processing 98% of oxygen we breathe. In each cell, there are hundreds of mitochondria. Each mitochondrion has several copies of mitochondrial DNA, which codes for 13 key proteins responsible for oxygen processing. Multiple mitochondrial DNA copies in each cell ensure the constant synthesis of these proteins, but in some cells, when mutated mitochondrial DNA become a majority, synthesis of ATPs is compromised and cells become respiration deficient. In humans, the process of accumulation of mutated mitochondrial DNAs to high percentages takes decades. First respiration deficient cells start appearing at around 60 years mark. In our twilight years, however, up to 30% of our neurons suffer from energy deficit, with all the consequences this development implies. Here comes the most relevant part. The type of mutations in mitochondrial DNA most often found in respiration-deficient neurons is large deletions, flanked by direct repeats. Deletions are a loss of a significant chunk of genetic material. Direct repeats are short, identical sequences in DNA, and the frequency of repeats in human mitochondrial DNA is rather high. Deletions form during replication or strand break repair due to the misalignment with the second repeat as opposed to the first. As a result, one repeat and all genetic information between two repeats are lost. The challenge we're facing is to eliminate or diminish the possibility of deletions in mitochondrial DNA. There are many possible solutions. The simplest and perhaps the most promising one is to modify mitochondrial DNA sequence in such a way as to reduce the number of direct repeats without violating the genetic code. The proposed solution is biologically straightforward because it does not violate status quo and therefore does not create additional logistical problems. There are other possible solutions for prevention of deletions, and some of them do not even require changes in mitochondrial DNA sequence. I intend to test efficacy of at least 3 interventions addressing different aspects of deletion formation. I ask for your help in solving this terrifying aging-related problem of dementias and potentially saving a third of our neurons from dysfunction and death long enough to keep our personhood intact to the very end. Also keep in mind that proposed interventions may help preventing aging-related muscle loss as well, because our skeletal muscles accumulate similar percentages of respiration-deficient myocytes. The minimum financial requirement for initiating these experiments is $50,000. The more you donate, however, the more I will be able to move the experiments beyond a proof of concept stage. Thank you very much.